Thank you very much for the invite to come and talk to you guys today. This is actually my kind of first non-marine conference. For many, many years, I was a marine microbiologist, and we looked at basically how viruses and bacteria interact in the oceans, and I still do a lot of that work. But COVID-19 really, we did a lot of sequencing with the COG UK group that's monitoring these strains, and that really kind of turned me on to this idea of your science actually having a real direct influence in people, and I kind of got addicted to that kind of input. So we set up the Citizens Phage Library as a result of that particular project. Thankfully, this is an audience that we don't really need to kind of introduce the real crisis that we're facing in antimicrobial resistance. This paper from Murray et al. In 2022 pretty much lays it out on the line that there are, I think, 5 million AMR-related deaths in 2019. That compared to about 6.5 million COVID-19 deaths that currently happen. And this is going to be happening every year and getting worse and worse. So we desperately need solutions to this problem, as you all well know. Now, the talk I'm going to be talking about today is primarily about compassionate phage therapy. Now, this is in the clinical setting, for those that don't know, is basically under Article 37 of the Declaration of Helsinki. If you have a patient that is no longer responding to kind of your standardized treatments of care, you can seek authorization to have kind of experimental treatments, as long as you frame that in a way that allows you to write it up as a kind of a scientific experiment, if you will, and capture all the details. So where phage therapy is typically being used, certainly in Western Europe more than well, certainly in the UK and in the States, less so in Belgium, is this idea for using it for compassionate phage therapy. A paper came out in 2019 where they looked at this, and they basically showed that it's still very limited, even though it was showing that there is great strength in this approach to dealing with antimicrobial resistance. There is actually quite limited to certain centers in the world. And part of that is the need for the availability of phages to become readily available and characterized and everything else. You may well have heard of IPATH. That's been recently set up in the States by Stephanie Strathdee and her group. And that actually shows even when you kind of have this structure in place, how few requests for phage therapy make it through to actually being something that you can put into a patient. So in here, they had 785 phage therapy requests, and then only 17 of 119 patients did it actually go into there. Now, part of the reason for that was, if you look in the data for this paper, it actually shows that in 99 cases, they initiated a phage hunt, but in 31 of those cases, the phage weren't found, and then in 10 of those cases, the patient actually died prior to those phages being found. So there's a major issue in terms of getting the phages, finding the phages in collections, and then if you can't, finding novel phages that can be used to treat those particular patients. Perhaps moving more to Europe, this is a kind of standard case, if you will, where there's a Chromobacter infection for cystic fibrosis. And if you look through the timeline of the kind of supplementary information from this paper, you can kind of lay out how long it took for each of these individual steps to go through. So you can see initially when they agree phage therapy, there's 23 days to produce that cocktail. And I believe in this case, they actually found phages in their collection that were suitable for killing the host pathogen that they were trying to kill. When they nebulized those phages and put it into the patient, there wasn't really a particularly good response to that patient. So they decided actually what they were seeing in vitro was not necessarily representative of what they were seeing in vivo. So then they had to launch a second attempt to try and find phages, and that actually took 105 days to get that. So taking a very long time to actually get the phages ready to get into the patient. So one of the thoughts we had is actually organizing field work and going out and doing these sampling campaigns actually takes quite a long time when you realize that you don't have the phages. So can you actually speed this up? If you just had a bank of samples, for instance, that were all arrayed in 96-well plates, that you could actually just very rapidly say, okay, we didn't have any phages in our actual collection. Can we go to our samples collection and look for novel phages for that particular pathogen? So that's one idea we had, that we could potentially speed this up. The other thing you might look at or see when you look at this is actually just moving phages across international borders, particularly when you're trying to get it into a patient, is a really, really long process. There's an awful lot of paperwork involved. And so what we really need is kind of nationalized or national centers that can do this. So rather than having to say import phages from from Brussels, and we can actually have our own capacity to do this. Ideally, those would all be interconnected in some way so that the paperwork is known to everyone, there's less bureaucracy, and the phages can frictionlessly move across borders. That would be the ideal. So even if you didn't have the phages in your national capacity, you could then rapidly bring them in from other places. So in the UK currently, there's really only been one recorded case of phage therapy, compassionate phage therapy, and that was the kind of poster child, as we kind of see it, as for phage therapy in the UK. And that was this Isabel Carnell Holdaway, who was treated for a cystic fibrosis infection, and with Professor Graham Hatfield, I believe, is talking tomorrow. And yeah, so apart from that, there hasn't really been a lot of kind of published work done in the UK. It seems to be quite limited. Part of that is definitely down to this lack of regulatory structure in the UK. There doesn't seem to be a huge amount of political momentum to implement one. Now, I do say that, but our new Prime Minister is about to be announced, and she's so hell-bent on deregulation, we might be able to inject anyone with anything by tea time. So we'll see how that goes. But yeah, I work locally with our Royal Devon and Exeter Hospital, and we have a patient there we actually have 4 patients, but one in particular is really, really suited to phage therapy. She would benefit from it enormously. It's a bronchiectasis patient. But the hospital really doesn't quite know what's needed to go through this process. And I think there are like 3 or 4 levels of ethics that you need to get through in that hospital. So there's no standard protocol for them to follow. So everyone's very nervous and risk averse for doing these kind of things. We also don't really have a dedicated phage biobank for this. At least we didn't, but I believe Martha is talking about this tomorrow, so that may well change in the next 24 hours, so watch this space. That should be very exciting indeed. There is kind of, I guess, anecdotal evidence, and you sometimes see it on these kind of Horizons reports on the BBC, that actually some patients that have kind of bronchiectasis will often go out to the Lava Institute and get treated privately. Now, obviously, with a universal healthcare like we have in the UK, it would be ideal if those kind of treatments were available to everybody. So I thought to myself, how do we best kind of design what would be an optimal system for phage therapy in the UK? And so I kind of came up with this idea of the Citizen Phage Library. And the main reason I think the good thing to reach out to citizens is for lots of reasons. But number one is you get to sample a lot of areas very, very rapidly. You can get diverse sampling from all over the country, be it in freshwater, from chickens and pets and bird baths and these kind of things. The other thing is you have this benefit of the commons, right? Your citizen scientists are actually actively engaged in the process. They're learning about antimicrobial resistance. They're actually feeling that they're contributing to the problem. So they're more likely to accept that kind of treatment, and they will build political pressure in order for that kind of treatment to be available to them. So that was kind of the aim of bringing this in the citizens. So what we essentially planned was we would send each citizen 3 vials that they could collect about 40 mil or that kind of same amount of soil or something in. They would use that What 3 Words app, which locates your GPS on Earth, and they would write that on the top of it, so then we know where those particular samples came from. And then they send them back to us for sampling. We then screen those against a kind of list of pathogens that we're interested in. And then we sequence the genomes of them, we compare them to see what their host ranges are, and then we look for kind of antimicrobial synergies and antagonisms with antibiotics that we have. All of this gets stored in a localized database. And then the phage hunter gets told that we found a phage in your sample, congratulations. They then get to name it, which they get very excited about, particularly the young kids. So very similar to the C-Phages program that Graham Hatfield launched. And then the idea is we have this database, and that hospitals would then be able to search through Phage Directory to say, actually, in the Citizen Phage Library is a particular phage that might be used to be treating this particular illness. So far, this is what we've done. We've gone out, we've done 6 school visits, 3 public outreach events, and captured around 497 samples. These have been done through kind of walkarounds with young kids, or going to school visits, or doing kind of Sidmouth Science Festival and giving them things.
We are looking for samples.
Okay. It's basically a kid saying what he's going to name his phage and everything.
Brilliant. And what is a phage?
Toby Toblerone phage.
It's a microscopic
Didn't like to say that was copyrighted.
Yeah.
So far we've got 337 phages isolated. So it's actually a pretty productive way of finding phages. These are against various hosts, so from E. coli, Pseudomonas, carbapenem-resistant Acinetobacter baumannii, Morganella, Proteus, Klebsiella. I'd probably say about 60% of our phages are E. coli, because that's what we were using initially just to kind of determine how many phages we would find. We've probably got about 80 or so Pseudomonas phages in there now. So we've really, this is in a kind of period about 12 months of heavy sampling that we've actually kind of collected all these things together. We've sequenced around 150 of them. That is really the current bottleneck that we're struggling. We dutifully extract this DNA and give it to the sequencing center at Exeter, and then it gets deprioritized, and other stuff gets done, and then we wait 3-4 months for the sequencing. We have got procedures in place. We actually want to bring this in and basically do it on the MinION sequencing using some of the approaches we use for kind of, I guess, my marine viromics type work that we did. And we think if we can get that work, I haven't got time to talk about it now, but we think we can get it down to around 10 to 12 pounds per genome if we would do it in-house on a MinION. So watch this space for some work coming out with that. Initially, the idea was that we would basically, people would be able to go online, order these kits, and then send them back to us. But actually, what we found very early on is that in the glass jars with the water that we were giving, and we chose glass because we were worried about phages binding to plastic, like some people have shown. Actually, the likelihood of finding a phage in those samples drops off pretty quickly if you don't filter them. And we suspect what's happening is those phages are just absorbing onto kind of solid material in that pot. And then when you spin it down, you're losing the phages that were in that particular thing. But what we did find is actually if you filter them immediately, then you can take a sample that's, I think the latest current one is 5 months is our longest, and then still get phages out of those samples. So what we developed, so basically it stopped us being able to ship these kits out to citizens. So we did it all in in-person events. But what we've now developed is this very cheap way of essentially just sucking the water straight through a filter into a syringe and then shipping that back to us. Again, we were slightly worried about phages binding to the plastic. So we did a whole bunch of tests where we tested syringe material, we tested the amount of SM buffer to see if that was stabilizing the phages, and different levels of tween that can stop phages binding to plastic. And actually what we found is it doesn't really make a whole bunch of difference. Almost nothing you do to it tends to stop a loss of around 10 to 100-fold of your phage concentration very, very quickly. Now, interestingly, Jessica Satcher recently put up a blog post on Phage Directory where they actually shipped Pseudomonas phages around Australia to see what the drop in titer was. And that was around similar, even if you're just sending the phages across the city. It doesn't seem to be related to time. So I think what we've got at the moment is currently a way that's essentially good enough. We can still get phages out of it, but probably still needs to be optimized. So if anyone's got any ideas about how we can potentially stabilize phages more in these sample kits, I'd be really delighted to hear them. Okay, so when we get these things in, essentially we just follow the Olson method that was published in Phage, where you just take this enrichment in these deep well 2 mil wells. You take your filtered lysate, your filtered sample, add in bacteria that you're interested in finding phages for, cultivate them overnight, and then filter them through a 0.45 micron filter, and then essentially repeat it in a much smaller volume of 200 microliters, and then plate them out on a spot assay. And at that point, you know that you've got phages from that particular sample. And then you go through the standard procedure of kind of doing dilution to extinction culturing to try and purify some of these phages out. So these are some of the phages we found. We found a phage we called Lemonade, or this was found, maybe this is back to carbapenem-resistant Acinetobacter. It's from the River Lemon. And actually, this was like 100 meters from my house in a small stream. We found it. We kind of bonkers that Acinetobacter phages there. Pseudomonas phage Cedric was isolated against the Swiss strain. And then citizen scientists actually sampled their chicken coop. And we asked them what they wanted to call the phages, and she called it Kylie Minogue, which was just genius. We actually found a second phage in there, so we called that Danny Minogue. So there are now 2 phages in that particular one. The obvious question to ask is when you're doing this, well, through enrichment, are we actually just sampling the same phage over and over again, right? So we took a subsample of the initially coli phages that we took from just around Exeter University and did kind of average nucleotide identity sequencing. And there are some similarities in there, but actually we are finding reasonable groups of clustering phages going on. And what we hope to do is actually bring some of the tools that hopefully Thomas is going to be talking about tomorrow in-house, so we can actually speed this process up slightly. Is it going to work if we actually have a patient come to the UK with this? So we tested this with a phage directory request that came from Switzerland. And actually, once they sent us the strain, within 3 days, we'd found phages for this particular pathogen within our phage collection that we'd all isolated. Interestingly, they're all jumbo phages, these sort of 250, 280 kilobase per genomes that showed these in vitro killing. By day 6, they've been sequenced internally on MinIRN sequencing and actually showed they didn't have any antimicrobial resistance genes. They weren't lysogenic in any way. So we thought they would potentially be suitable for treatment. And then we had a week where we could actually train them up. And hopefully you can see on the bottom that once you do train these phages just by infecting sort of naive host over and over again, you actually develop these things to reduce the emergence of resistance in the host. The second one, we actually had a patient, as I was talking about briefly in the RD&E. We worked with the Environment Agency. The Environment Agency are based maybe about, I don't know, 20 miles away from where we're located. And pretty much all of the sewage samples for COVID-19 sequencing were actually going into the Environment Agency there. So we said, well, can we have a milliliter of each of those samples? They said, absolutely. So we had this huge screen of wastewater samples. And against this particular strain, we found 63 phage or positive hits against that particular strain, which was slightly problematic in that how do you actually then prioritize which ones you want to take forward for treatment? So we worked on a way of figuring out how we can do this with image analysis of our plaques. So what we really want to prioritize is fast growing and halo plaques, and then deprioritize any plaques that grow slowly, potentially lysogenic through cloudiness, or have actually this kind of evidence of rapid emergence of resistance by seeing these kind of, these texture plaques that you get. So what we do is we essentially have 3 HP scanners or Epson scanners, I think they are, in a controlled environment. And then we scan them over time as the plaques develop. And what you can see is the plaques emerging on the right-hand side. And then we do a differential image. This is with, so Remy Chait does all this stuff at Exeter. He's a wizard, if you ever want to work with anyone who does any of those sorts of things. And so then when we do that, we just simply borrowed the 1 Petri neural network to identify plaques on your plate, and then use ring detection within those boundaries to determine how the plaque was growing over that particular time. And what we actually found is you can start pulling out some cool features from these plaques, right? So even though your plaque radii are actually quite variable on your plate, actually the growth rate is quite constant once you fit a polynomial model to those growth rates. So you can use that to actually kind of start to have a proxy as to whether or not your plate is now axenic, because all of your growth rates will look the same. So you can get all of that information in real time. The other thing is you can actually do feature extraction simply by pulling a mean radial profile through your phages. So this dotted line on the left-hand side is actually kind of your background of your lawn. And then you can see how as you go into the plaque, the intensity of the light coming through, or goes the other way, sorry, changes. And you can start to use those things actually to determine the types of things you're interested in. So bullseyes have an interesting feature whereby the boundary of the plaque is actually slightly darker than the lawn itself. So it suggests there's growth being promoted in the bacteria at those particular points. You might want to avoid those, but then you can find ways to isolate phages that make these nice halos. And then we're working with Singer Instruments to basically take these and then get them picked so we can get them into libraries very, very quickly. Finally, the one thing I want to talk about is essentially what we want to do is capture everything we're doing in these phages. So essentially a living record. So rather than just NCBI, also have all this data. So, if you do a kind of, if you do a PFU count under a particular condition, you capture that information. If it goes under, different types of growth and different types of hosts, you can capture that particular information. And now there's lots of databases where actually they're not relational. You can just keep adding stuff to the records. They're very, very flexible. And we use CouchDB to do that and capture this particular information. So for instance, you might have a section of that is actually just the provenance of it that would be kept forever with the genome. And then you might have a processing step. So every time you do it, you capture a particular process, the method using the DOI and the results of it. And all that would get basically added onto this virus whenever it was used. It allows you to distribute these phages because you can actually just send them out and say, on this particular day, I sent it to this particular person. And then whoever checks this record knows exactly where that phage has been distributed. So it really promotes sharing of the phages. But you can really imagine the strength of this once you start adding in clinical trial data and when it's been used in compassionate use and how it responds to the immune system and these kind of things, and building all that in so that actually once you do have this kind of big record of data, then you can just point data mining algorithms at it and start really pulling it out to understand some of the features of phages that make for really beneficial analysis. So this is currently all being done on CouchDB. The end goal is actually to put all of this on a blockchain that would allow it to be this kind of immutable record that would really kind of fix who discovered that phage and make it publicly searchable. Blockchains essentially are behind Bitcoin, but they can be used for good as well as evil. And they're essentially just a fancy NoSQL database that gets propagated. So these are used for actually moving seafood around now, because you get these smart contracts generated at each stage that allow for the generation of kind of billings of lading and material transfer agreements could potentially be generated through smart contracts to really speed up this kind of frictionless movement of phages. The aim of the Citizen Phage Library ultimately is essentially have a collection of phage libraries around the world that are interconnected and can share data between them. And the first one we've actually set up, if the funding gods are kind to us, is we're hoping to basically roll into the next round of the Phage Australia funding. So they'll be setting up the first Australian branch of the Citizen Phage Library, and through that we'll start working out some of the, I guess, the barriers to changing these things between different countries and sharing them across those. So that's where we are currently. So that's the ultimate aim, is a network of cost-effective, connected, citizen-led projects providing on-demand, fully characterized phages for any bacterial infection anywhere. If you're interested in the project, please go to the citizenphage.com website. You can register on there, and hopefully with the new syringes come out, we can then start sending some kits out, and you can get involved in the project and start finding phages to help us. Finally, I'd just like to acknowledge everyone in the team. So many people have jumped on board of this as kind of volunteers and helping things out. My team has been amazing for helping get this working. Extra Science Centre have been driving all of the kind of outreach stuff, and they've been amazing. So yeah, thank you very much.
I'm Rob Levine from Belgium. The stability issue, right? Firstly, I think in general it's perceived that high titer phages tend to be more stable. So I think the initial titer of your is going to be critical in the amount of reduction you have. I think that's the first thing. But secondly, as I understand it, it's still at the isolation step. So do you care about the log2 reduction?
So ultimately, no, as long as, if you lose 99% of your phages, and there's still a lot of phages in there that might infect your particular pathogen. My only concern is for rarer taxa, you might actually lose the few that were in those samples. So yeah, optimizing it would be nice, but not critical going forward.
Okay, thank you.
Great talk. I'm Ville from University of York. So I was thinking, have you looked, classify the different phage plaques based on how they grow on light spears? Have you tested? Is there any have you done analysis? Is there certain type of plaques that are having higher host range or more effective, or can you how much can you generalize based on all the data you're generating?
Ultimately, that's definitely what we have on the cards. We've just got all the sequence data back from there, so then we can start comparing it to the plaque sizes. But yeah, if you could ultimately use those plaques just to say, actually, we think this is this particular type of phage because we've seen this type of growth rate previously in this morphology. Obviously, there's going to be limitations in that depending on how thick your agar is probably going to change it, and fluctuations in temperatures in your growth cabinet might change it between runs, but it'll definitely give you a good first pass to just simply reduce the search space.
There's a question from Janet Nail.
Thank you. That's Janet again from SRUC. Interesting talk. I was wondering whether the platform will be able to record unsuccessful treatments or resistance to the different phages, and how would you be able to overcome those? Thank you.
So it'll definitely capture failure. I think that's a critical part of determining which phages you want to be looking at in terms of the future things. Certainly in terms of antibiotic interactions, which ones are antagonistic and synergistic. I think all that information should be captured on there. If you're testing a phage against, say, 3 or 4 different, or 50 or 100, depending on your isolate collection, different types of Pseudomonas, then every single one of those should be in there, whether they succeeded or not, and what the titer was, the efficiency of plating method. So pretty much anything you could do to that phage, as long as you can record it in text in some way, then you could add to the phage record so that anyone could come along. And if your ontology is kind of defined, then people can then use computers to search that space to find out what the answer was to the question you had.
Hi, nice talk. I was wondering if you use always the same strains for isolation, or you change them?
So we typically isolate from citizen science samples on, we have a BW25113 E. coli, and then we do a PA01 Pseudomonas, and then we, if we have a project where people need Klebsiella phages, we'll add in a Klebsiella to it. The RD&E strain of Pseudomonas we have seems to be quite promiscuous from early data, so we're probably potentially switching to that from PA01. We're just about to start doing the test, but certainly when it comes from citizen samples and we're isolating phages to go into the phage bank that we already have pre-isolated, then yes, we use a defined library for those.
Just one in the interest of time. One more, it's very quick.
Just so the plaques are done with single phage on a bacteria, do you multiplex up the phage then to kind of try and kind of increase the power or the kind of killing power of your phage?
So you mean make phage cocktails out of them?
Yeah.
We haven't currently done that as yet. We have a system we're testing whereby you can measure combinations of phage, and whether you see, a bit similar to a talk earlier, whether you see adding the phages together or adding them sequentially, actually do it. And we do that using some swim assays that we're currently working on. So you can just line up the phages and then drive bacteria through them as they swim, and then see whether you get emergence of resistance as it goes through this wall of phage, which is, again, it's Remy being a wizard doing this. So yeah, that should come up fairly soon.
Thank you very much. Thank you. That's great insights into those collections and efforts that you're doing there. So thanks once more.